A good morning or afternoon to you. Thank you for joining us today. I'm Jan Reichert, Executive Director of the Antibody Society, and I will be your moderator. Nick Hutchinson of Fujifilm is here also as a moderator. Today's webinar is one in a new series designed to inform and educate our members, as well as the broader scientific community, about topics relating to commercializing antibody therapeutics. Our speaker today is Dr. Christine Power, who is an expert in business development relating to antibody therapeutics. Via her consulting company, Dr. Power provides scientific advice and scientific opinion in the field of antibody discovery and development to biopharmaceutical companies and universities. Today, she will tell us about scientific evaluation and due diligence of innovative monoclonal antibody therapeutics. Please note this webinar is being recorded. Slides can be downloaded from the Materials tab in the viewer. Please do add any and all questions to the Q&A box in the viewer, and Dr. Power will answer those at the end of the webinar. Without further ado, I'll now turn the show over to Dr. Power.
Hello, everyone, and special thanks to Nick Hutchinson and Janice for inviting me to give this presentation today. So I'm just going to start with a disclaimer. The views and information or opinions expressed in this presentation are solely mine, and they do not necessarily represent those of my former employers. Okay, so I'm first of all gonna, I have a bit of a problem forwarding these slides here, if you just bear with me. Okay, so in today's presentation, I'm first going to discuss the importance of in-licensing to the pharma and biotech industries, and then how we actually identify the in-licensing opportunities. I'll then go through what the due diligence process actually entails, and then I'll give some examples or case histories of some that I've experienced personally and other examples from across the industry. And then finally, there should be a few minutes left to answer some of your questions. So over the last 15 years or so, I've been involved with over 70 evaluations and due diligences of both molecules and technologies. And so on this slide, I've just listed some of the more interesting opportunities. I am actually not going to go through them. So a recent analysis from Innova and Eli Lilly revealed that approximately 80% of the FDA-approved medicines in the last 10 years were not actually registered by the originator or the patent assignee. Now, many novel therapeutic molecules and technologies are actually initially conceived and developed by small biotechnology startup companies or universities, but it's usually big pharma or biotech companies that will bring the resulting therapeutic to the market and commercialize the product. So recently, there have been a trend for large pharma companies to reduce in-house R&D activities and increase in-licensing or acquisition of products to fill their pipelines. And so this is particularly in view of expiring patents or the so-called patent cliff that many of you may be aware of. So new technologies are also being developed rapidly. So it's more feasible for pharma companies to rely on startups for initial drug development in order to acquire de-risked programs at later stages in development. So once a potential asset has been identified, it's necessary to perform an in-depth scientific evaluation and due diligence in order to increase the probability of a success before finalizing any deal. So in this presentation, I'm going to describe what this process entails with some case studies and some of the pitfalls that may be encountered. So the path to a marketed antibody has often been likened by some to climbing Mount Everest. It's actually a long and arduous and expensive process, and many drugs fail to make it to the top. So most of you have probably seen various iterations of this slide. On average, it takes about 10 to 12 years for an experimental drug to traverse from the laboratory setting to the clinic, to the patient. And most of the time is actually taken up by rigorous testing in clinical trials, which is actually necessary to minimize any chances of the drug causing harm to the patient. So developing a new prescription medicine that gains marketing approval is estimated to cost drug makers between about 1.3 billion and even up to 2.6 billion, according to a recent study performed by the Tufts Center for the Study of Drug Development and recently published in the Journal of Health Economics. So the cost actually increases dramatically as the drug progresses through clinical trials. So the average cost of a phase 1 trial is about 3.4 million, and this increases to 8.6 million for a phase 2 and estimated to be about 21.4 million for a phase 3. And obviously, many companies perform several phase 3 trials in order to before registration and approval of the drug. So this is obviously why it's so expensive. Now, okay. So given that the drug development costs increase substantially as the therapy moves into the clinic and then to larger clinical trials, most companies are actually interested in making business deals during the early phases of clinical development, where there is evidence of clinical activity that reduces the risk and the licensing deals are more favorable for the acquirer. So access to assets already in clinical development can drastically reduce the time to market. Now, failure in phase 3 is actually catastrophic for both patients and the company, because up until this point, the company's actually spent hundreds of millions of dollars on the drug discovery and development process. So in a recent article from Manuel Hermosilla, who's at Johns Hopkins University, he noted that there is often a rush for interim licensing deals after a phase 3 failure. However, quickly executed deals can often result in failure. So the majority of innovative biotechnology ideas can be traced back to basic research that began at an academic or research institution over the course of many years and possibly even decades. Now, if you examine the original source of most successful biotechnology products, so as I mentioned earlier, you'll find that the majority of them originated from basic research, which was conducted by scientists, professors, physicians, or even engineers at an academic site or research institution. Nonetheless, very few products have been fully developed within academia and research institution. And this is because their goals are actually not the same as in industry. And they possess limited experience in moving product concepts efficiently to product development and commercialization. So in this slide, I've limited some of the, listed some of the reasons why universities or startups want to form alliances or outline to big pharma. Now, interestingly, universities may choose to hand over their patent rights on a discovery to big pharma companies for a one-off payment rather than receiving royalties later. And this is particularly useful because it can fund their ongoing research programs immediately. So before entering into a licensing deal, universities and startups need to prepare well in advance for the due diligence process. They need to expect to be able to answer detailed questions, and if they are actually unable to disclose something, explain why. So what is due diligence actually? So due diligence is the thorough review and assessment of a potential asset to determine the asset's viability to get to market. Now, due diligence involves a detailed analysis of the available data, relevant licensing decisions and negotiations, and it includes looking very detailed level at reports, literature, and even the competition in order to guide these decisions. So a robust due diligence process requires diverse scientific and regulatory expertise in order to ensure that an accurate and holistic evaluation of a potential asset is taking place. So a positive outcome would be a mutually beneficial partnership founded on trust and transparency. So how are the opportunities identified? Well, search and evaluation groups identify early opportunities in the fields of their strategic interest and portfolio fit for their company, or they may be looking for areas that the company would like to move into. So for example, in rare diseases or infectious diseases. So this is done through targeted and regular attendance of partnering and scientific conferences, through scanning of new and recent literature and patent databases to identify emerging opportunities, and searches of grants, deals, and pipeline databases. However, using the search, it's important to use the right search term, for example, because that can make the difference between finding a novel asset or nothing at all. And not every search term will yield success when searching across multiple databases. So outreach as well to networking with university tech transfer officers, key opinion leaders, industry contacts, and licensing professionals is also a source of opportunities. Or there may be a direct approach from startups or university researchers or their technology transfer officers to present their assets. So the ability to seek out potential in-licensing opportunities or partners with the most promising assets under development can bring huge rewards to the company, but getting it wrong can lead to severe financial and reputational implications. So once a suitable asset has been identified, the next step is gaining access to those assets, either through licensing, a joint venture, or even an acquisition. Now, the due diligence is a critical review of the entire data package and hypothesis, the scientific rationale from the target through to the mechanism of action, the clinical relevance, and the evolving risk-benefit balance in the context of the available preclinical and clinical data. So it can take considerable time to identify and explore potential issues, but usually takes between several months and 1 to 2 years, depending on the development phase of the potential asset and the urgency of the company to fill the pipeline. So this slide outlines the main steps in a due diligence process. I'm not going to go through this. You're all going to get the presentation, so you can go through that at your leisure. But I first wanted to say that startups usually provide a non-confidential overview presentation of their asset after signing, and then after signing a CDA, a confidentiality agreement or a non-disclosure agreement, they will provide more extensive information to the company. So this is usually followed by a face-to-face discussion where the asset can be discussed in more detail by experts from either side, and may even include a site visit, for example, when a technology is concerned. So management approval is normally required to advance to the next stage of the process, and the process can be stopped at any point depending on the findings. So, for example, the budget is not approved by senior management. So it's absolutely crucial to set up a data room during the due diligence process in order to exchange confidential information, such as raw data and reports, rather than attaching files to emails where they could be lost or intercepted. And then it's not until later stages that financial details are negotiated, and these are for upfront payments, milestones, and royalties. So the first stage in the process is to identify an asset. So what are we actually looking for? So first of all, potential strategic fit of each unique opportunity. The molecule should address a novel disease biology and target a protein with first-in-class potential. The molecule is already a lead candidate, preferably in the lead optimization stage, or better still, the molecule is even more advanced and has demonstrated efficacy in disease-relevant cellular and, depending on the stage, and in vivo models. And the best case scenario is that the molecule is actually IND filing ready. So pharmaceutical companies are not only in licensing molecules to fill their pipelines, but they also face an increasing need to update and upgrade their R&D capabilities if they want to sustain the development of their pipeline and continually bring new drugs to market. So within licensing, companies can promptly tap into and use ready-made external R&D solutions. So this process not only saves time, but also resources that would have been committed to developing solutions in-house from scratch. So using technology licensing, firms can be in a unique position to directly and quickly respond to technological innovations. And when looking for new technologies, I've just given a list here of some of the things that we're looking for. So for example, is the technology cutting edge? Will it open up the target space to us? So for example, access to targets that are currently undruggable with antibodies. So for example, intracellular targets. Is it proprietary? So what's the IP situation, and have the licensors actually infringed others' patents? Are there any related or competitive technologies? Has a landscape search been done to see if this is the truly best technology available? Is there a sound scientific basis? And then actually, is it transferable? Can it be used in-house? Will this technology actually work? Does the startup have any proof of concept studies? And of course, look at the strengths and weaknesses. And then most importantly, is the research idea novel and the technology interesting enough to be funded? And is it a platform technology that could have multiple future applications, or is it just going to be a one-off? So who's involved in the due diligence process? So due diligence experience, scientific expertise, and strong industry background are really required in the due diligence team so that they can properly gauge the opportunity and understand what effect any issues that are uncovered might have, and also be able to properly root out these issues. And these would depend on the stage of the asset. So the business development team usually identifies expert consultants in research. So these could be immunologists, protein chemists, preclinical development for in vivo studies and so on, clinical development, CMC and supply chain, so this is the manufacturing feasibility, regulatory, market and payer research, and most importantly, intellectual property. So in some ways, a good due diligence is really like a detective's investigation, if you may. And in order to be able to do it effectively, it takes experienced professionals who have a wealth of past project experience to draw upon. So it's not as simple as just going through a checklist. So though the due diligence process can be tedious and time-consuming and sometimes even very frustrating, it's a necessary prerequisite to a well-planned transaction. So depending on the stage of development, the key questions asked during a licensing evaluation will change. Once an opportunity has been identified, I've listed here some of the questions that need to be answered during the due diligence process. So first of all, does the asset address an unmet medical need? So what I mean by unmet medical need, it's where the current treatments for a condition or a disease are insufficient. So are there supporting scientific data? And are those data sufficiently compelling? Is there sufficient differentiation from competitors? Is the product candidate proprietary? So is there intellectual property in place? So this is important for exclusivity and freedom to operate. These both hinge on the intellectual property rights. And patents and know-how each provide market exclusivity, precluding competitors from taking market share for a certain period. And then, can the asset be manufactured? So moving on, is the project or business truly in the condition that is being presented by the potential licensor? Have any corners been cut? Are there any gaps? So what are the risks of taking this on? And are there risk mitigation strategies in place? What is the pathway to market and to value realization? So in simple terms, basically, how long will it take and how much will it cost? And then, is there a scope for a killer experiment to yield a definitive go or no-go decision? And then finally, does the startup organization have the right team to actually do the job? So, some of these startups are a very small number of people, could be a virtual startup, could be only 9 people or something like that. So can they do the job? So this is a very detailed slide, and I don't really expect you to read it. But in it, I've listed some of the key data which is required for the due diligence process. As I mentioned already, it depends on the stage at which the asset is planned to be in-licensed. So it's going to take me too long to go through the slide, but I just wanted to remind you that the presentation is going to be made available later. And if you want more details, you can find it in an excellent recent article published by Dolly Paras-Sampuria in Drug Discovery Today, and the reference is given at the bottom. So just focusing on important considerations at different stages in development. So for early stage preclinical assets, the questions are generally related to formulation, developability, receptor occupancy, target engagement of the antibody, acute and long-term toxicity, preclinical assessment of metabolic pathways, tissue distribution, translatability, and projected dose. So a crucial element of any assessment from the clinical perspective is the formulation and the route of administration. So changes in formulation or route of administration or manufacturing process, indeed, for both small and large molecules, so biologics, can have a dramatic effect on the PK of the drug, and that can then have a downstream impact on safety and efficiency. So if different formulations are used, for example, in different preclinical and clinical studies, the differences might make it difficult to pool and interpret data. And so for this reason, it's actually important to ensure that the same formulation has been used for all patients in the clinical study. And likewise, if there's been a manufacturing change for a biologic, it can result in changes in the anti-drug antibody formation, ADAs, and that can in turn affect the PK, safety, and efficacy. So, where animal models can provide meaningful data, the design of the first-in-human clinical trials can be optimized. But in cases where preclinical data are limited, so there are no animal models due to lack of the antibody cross-reactivity with its ortholog, or transgenic models are not available. So actually, this lack of cross-reactivity is not unusual, because monoclonal antibodies are particularly have very, very high specificity for their target. So often the first-in-human studies would be the first time to be able to generate safety data through the clinical trial itself. So a phase 0 trial might be performed in a limited number of subjects at a low dose to gain understanding of the mechanism of action, biodistribution, tumor targeting, PK, or to guide selection of a dosing route. And in fact, in oncology clinical trials, this is possibly this is normally done at phase 0. So then moving on to key questions in clinical development, This would concern whether the relationship between the target phenotypic assay and disease has been established. How does target expression vary across tissues? Have target-related and off-target safety margins been established? Can an exposure-response relationship be generated in vivo to demonstrate proof of concept? And then can biomarkers of each stage of the exposure-response relationship be generated to measure this both in preclinical and early clinical testing? So if they haven't been generated, they really need to be generated early on in clinical testing. Now, is the indication chosen appropriate, and does it offer the fastest path to prove efficacy? And then finally, does the trial design answer key questions and give a basis to continue the development, if successful? So all in all, this means, does the therapeutic hypothesis work with meaningful clinical benefit? And then finally, are there any safety concerns thrown up which are sufficiently understood and able to be managed to an appropriate risk level? So in terms of clinical trials, for due diligence, it's really imperative to assess whether clinical trial data are representative of the patient populations from the perspective of demographics, genetics, and molecular determinants of therapy, to ensure that the right patient and right dose for that patient population has been identified. It's also particularly important to, as I mentioned in the previous slide, that appropriate diagnostics are being developed for enabling precision medicine. So to identify patients who are most likely to benefit from the novel therapy, and also for stratifying patients to the appropriate dose and dosing regimen in clinical trials. So due diligence findings, they can actually also be negative. For instance, it can reveal that there are commercial and market access issues. There's no differentiation from competitors. There are potential CMC manufacturing issues, negative risk benefit. And, you know, despite what's been presented, there is insufficient data. It might reveal there's insufficient data for IND filing. So also there could be a disconnect between development plans and commercial timelines, and then negative KOL opinion sometimes, so key opinion leader opinion, regarding the use of the asset of the drug in specific geographic locations. So I'm going to shift gear now and give a few examples that I've personally experienced, and also some more famous examples. And probably one of the most famous examples of a successful licensing deal is actually the story of Humira. So back in 1991, the Cambridge Antibody Technology Group was a spin-off from the Medical Research Council, and they had developed phage antibody library technology for the discovery of human antibodies. And you may remember that Greg Winter received the Nobel Prize for this a few years ago. Now, in 1994, BASF, which was a German chemical company, signed a collaborative a research agreement with CAT to access their patented phage display technology. And then using the CAT technology, BAS made a fully human version named D2E7 of a mouse monoclonal antibody that was generated against TNF. So this antibody entered clinical trials and had really interesting, very good results from a phase 1 trial in RA, rheumatoid arthritis, involving about 140 patients in 1998, and then a second phase 2 study on about 283 patients in 1999. So phase 3 trials were initiated in 2000. So by this time, it had generated a lot of interest in the pharma industry. So Abbott acquired BAS Pharma in 2001. They did further work in this area, and Abbott submitted the biologics license application to the US FDA for the antibody, which is now called adalimumab, and also submitted to the EMA for approval for marketing for rheumatoid arthritis. So at the end of 2002, the antibody was approved for treatment of rheumatoid arthritis, and has since gone on to be approved in numerous other autoimmune indications. And so it's raked in billions of dollars for Abbott, and of course resulted in their royalty payments to CAT. So subsequently, CAT was acquired by MedImmune, which was then acquired by AstraZeneca, hence the shark eating the tuna, eating the smaller fish analogy. And this acquisition resulted in a flurry of activity by top pharmaceutical companies to get access to antibody display technologies. So I'm going to give another more recent example now, and this is the acquisition of Tesaro by GSK in 2019. So in 2015, GSK traded its oncology pipeline with Novartis's vaccines business. But by 2017, with a new CEO and CSO at the helm, the company did a U-turn and went back into the immuno-oncology therapeutic area. But they had to in-license assets to build up the pipeline. So they acquired this company, Tesaro, and it turned out to be a very astute piece of business. Tesaro, based in the US, their pipeline was sourced through business development partnerships, collaborations, and in-licensing of oncology assets. So they were able to identify unique oncology assets and subsequently acquire or partner on the development and commercialization of those assets. So when they were acquired by GSK, their pipeline comprised of an FDA-approved PARP inhibitor called Zejula, and they also had a number of drugs, 1 in phase 3, 5 in phase 2 studies, and 3 additional novel drugs in phase 1 studies, plus several more bispecific antibodies in preclinical development. So it really was an astute piece of asset scouting and business deal of significant value for GSK. So now I'm just going to give a couple of examples from my own experience about problems which can arise after or in licensing, or basically after a deal has been done. So they are perhaps a little bit dated, but I think they show the importance of doing a thorough due diligence. So As you've heard, antibody phage display or display technologies are the most widely used in vitro selection technologies used today for antibody discovery. So back in the day, so this is in the 2000s, from 2003 to 2010 and onwards, access to antibody phage display was controlled by a few companies based on their technology patents. Now, we wanted to in-license the technology, so Serono did an evaluation of a number of antibody companies, including CAT, Zoma, Morphosis, and in the end, they decided to in-license the Diax Fab phage display technology. Now, this was quite a successful deal, as well as the technology using the technology in-house. Serono initiated a number of funded research programs to be run at Diax, and one of these ended successfully with the discovery of avelumab, so it's also known as Bavencio, and this is an anti-PD-L1 antibody, a checkpoint inhibitor antibody, which was first approved in 2017. So, what was the unanticipated problem after in-licensing? It was gatekeeping. So, the development of antibodies using the Diax technology was subject to gatekeeping. So gatekeeping is a process instigated by the display companies, and it's a process that determines if you are able to pursue development of antibodies against a nominated target. Now, it was a way for the phage display companies to keep targets to themselves or their parent companies in order to avoid competition or prevent different licensees working on the same target. So, target exclusivity. Now, we knew that there was going to be a gatekeeping process, but the impact was unanticipated because there was no way to see what targets were on the restricted list. And so, as it turned out, by 2009, a number of Merck Serono's nominated targets had failed the gatekeeping process. So we had to put a mitigation strategy in place, and that was to license a new single-chain FV phage display library, and that was done in 2009, which was free from target gatekeeping restrictions. So the next case I'm going to talk about is generating functional antibodies against GPCRs. So GPCRs are multi-membrane spanning protein, and there are more than 800 members of the GPCR family that have been identified so far. And sales of drugs targeting GPCRs account for about 27% of the drug market. So these are GPCRs are a cornerstone in cell signaling. They are the transmission signal from the external environment of the cell and translate to downstream signaling cascade, which leads to the cellular response. So they're actually very, very interesting and attractive targets. But pharmaceutically, even though they're very important, so there are about 30% of all small molecule therapeutics target GPCRs, they're notoriously difficult to work with. So in the next slide, I've just outlined the progress in the development of generating antibodies against multi-spanning membrane proteins. So if they were originally targeted with small molecule drugs, why would you move to antibodies? Well, the reason is that the specificity of antibodies offers tremendous advantage when targeting specific GPCRs, they have a lack of off-target effects, coupled with low toxicity and high affinity, so longer residence times. And this makes antibody therapies even more appealing. So in addition, they can be optimized for effector function and serum half-life. Now, you can typically screen about 10 billion antibodies from the libraries currently available, whereas for small molecule drugs, even the most efficient high-throughput screening systems can only screen a few million at a time. So this slide shows anti-GPCR antibodies in development, and it was taken from a very recent review by Charlotte Hutchins. And this really shows that there is an increasing interest in GPCR as antibody targets, and this is probably due to the advances in the technology for their discovery. So in 2010, there were about 15 programs that targeted about 10 different GPCRs. But now, at the time of this analysis in 2019, there were over 170 active programs addressing 76 targets, which virtually doubled the activity in the area compared to 2017. Some of the antibodies, which are evaluated for more than one disease indication, and they could be at multiple stages of development. So this next slide just outlines why it's technically difficult to generate antibodies against GPCRs. So essentially, they're poorly antigenic. It's difficult to purify the antibodies to homogeneity whilst maintaining an active conformation. Cellular immunization strategies are hampered by the fact that only 10 to 20% of the surface is exposed, and expression levels of the receptors are generally low. So although there has been some success using extracellular domain peptides, these antibodies very rarely recognize are very rarely functional agonists or antagonists of the native receptor. And again, the high degree of sequence conservation between humans and rodents means it's difficult to break tolerance. But finally, they have a rather complicated biology. They exist in multiple conformations in active states, as well as having redundancy and ligand receptor pairings. So it's, generally difficult to make functionally relevant antibodies against these receptors. So the case I'm going to talk about is, again, it's a fairly old case, but we wanted to generate antibodies against GPCRs, and we knew the importance of maintaining the structure to generate functionally relevant antibodies. So this was done at a time when DNA and immunization technologies, which are widely used today, was in its infancy. So we evaluated a number of different technology platforms shown on the previous slide, and we decided to select MSM Protein Technologies, which was a startup based near Boston, for due diligence, and they had developed a proprietary paramagnetic proteoliposome technology. So the advantages of this technology were they overexpress the antibodies in cells, and then they developed a proprietary method for purifying the antibodies from the cells, and these were then incorporated intact into the paramagnetic proteoliposome particles, which displayed the GPCR in the correct orientation in its native state, highly pure, highly concentrated, and retained its ligand binding characteristics. So, at first, after in-licensing, things did not go according to plan. So MSM was a small company. There were about 9 people working there at the time. And one of the key members of staff who performed the necessary screening, who had the necessary screening experience, left the company just before the program started. Then the next problem that we encountered was the fact that when we licensed the dioxphage display library, we discovered that we weren't allowed to transfer it to a third party for screening. So all of the screening work had to be done at the Merck Serono facility in Billerica rather than at MSM. And then MSM wanted to keep their know-how on the preparation of the PMPL secret. So it ended up with the particles being prepared at one site and then transferred to the other site for screening. So it ended as a 3-way collaboration between MSM and EMD Serono and Merck Serono in Geneva, where all of the cell-based assays and engineering was done. Nevertheless, the collaboration ended successfully in 2012 with the identification of a lead candidate that met the TPP criteria that we established at the outset. And in fact, a patent on neutralizing CXCR5 antibodies for treatment of MS was granted in 2017. So another last case history is, I'll go through this very quickly because I realize that we're running short of time. And I've called this "What's Wrong with My Antibody?" So it's interesting that about of 131 mAbs currently approved, 82 of them are humanized antibodies, and the high number probably reflects the fact that the immunization and hybridoma technologies were mostly used for antibody discovery before the advent of in vitro display technologies and transgenic mice expressing human antibody genes. So one of the pitfalls of in-licensing opportunities is that they are based on activity of mouse antibodies against a target of interest in relevant disease models. However, to progress these antibodies into clinical development requires humanization and optimization, and that's not so straightforward as it turned out. So while I was at Serono, we had a long-standing collaboration with the Weizmann Institute in Israel, and they had funded a number of successful research programs over the years. And during one annual review, our interest was piqued in an immunization technology used to generate potent antibodies against MMP called a zinc tripod immunization strategy. So one of the anti-MMP antibodies that they identified was of particular interest because it was shown to be active in mouse models of inflammatory bowel disease and multiple sclerosis. So these were therapeutic areas in which Merck Serono was active. So we initiated an in-house evaluation. So the recombinant antibody was produced in HEX cells after the VH and VL cloning had been done from hybridoma cell lines. But in a nutshell, something very unexpected happened. The recombinant antibody could not recapitulate the binding activity of the purified antibody from the hybridoma. So once we'd seen this, we couldn't go back. So obviously, it's not feasible to continue development. Once we'd seen this, it's not really feasible to produce antibodies at an industrial scale from a hybridoma. We needed to have a functional antibody. So after doing a lot of digging through reports, lab notebooks, and so on, we decided to terminate the collaboration because we thought it was not going to be easy to resolve this problem. And in fact, there was a publication that came out by Babrak et al. In 2017, and it concluded that the simple expression of the correct VH and VL does not necessarily ensure a functional antibody, as there are contributions from other absent immunoglobulin sequences, post-translational modifications, or secondary or tertiary structure that may impact on antigen binding activities. So finally, my last example is a more recent one. So in terms of licensing, things can happen pretty fast when needs must. And in this case, it was the pandemic. So the outbreak of COVID in 2020 started a rush to develop antibody-based drugs, primarily led by big pharma and biotechs. Now, Regeneron and Eli Lilly, large companies which were already well versed in making antibody drugs, were the first to have engineered antibodies targeting SARS-CoV-2 approved. So these companies have moved extremely fast and used adaptive and streamlined trial designs and other measures to accomplish what usually takes years in months. So GSK were a little slower to get on board the antibody bandwagon, but from March to May 2020, they received an unprecedented number of collaboration proposals for antibody-based and small molecule drugs, vaccines and technologies that could be developed for treating COVID or be useful for future outbreaks. So in fact, they signed a deal with biotechnology, a small biotech company, to develop antibodies against SARS-CoV-2. And they also made an equity investment of 250 million to Vir. So the result of this was sotrovimab, so also known as VIR-7831, a monoclonal antibody used in the treatment of COVID-19, was granted emergency use authorization by the US FDA on the 26th of May 2021. So the greatest challenge is not in finding assets available for in-licensing, but sifting through the mountain of available assets to discover which ones meet the company criteria, are unique, and are based on sound scientific work. So finally, my last slide, just a few conclusions. Due diligence is a fundamental and necessary exercise prior to starting any in-licensing of a functional molecule, of a molecule or a technology, or collaborative product development, and should be an ongoing process. It requires sufficient expertise and experience and a manageable process and timeline. You need to identify any potential landmines and have mitigation plans prepared to sidestep them. And if the due diligence reveals that a product or a technology is unlikely to succeed for any reason, it's better to cut one's losses and stop. And then finally, for any deal to be truly successful, both sides need to show trust and transparency and come away from the negotiating table with a sensation of a win. So thanks for your attention. And I've just left, I'm happy to answer any questions, and there are my contact details if you wish to contact me privately.
Thank you very much, Christine. Thank you for an excellent and very clear presentation.
I'm sorry I ran a bit over time there.
Oh, that's not a problem. We have a few questions from the audience, so let's make a start if that's okay, Christine, and we'll try and answer some of these. So the first one, are there any due diligence software that companies use to evaluate in-licensing opportunities? And if yes, what are the factors or inputs that this software relies on?
Yes, so there are a number of private companies which do provide sort of due diligence software. So essentially, they're more or less databases where you can actually track the progress of the due diligence process. So incorporating all of the data. So they're like data rooms where you can put all the data, and they essentially track the dates of when meetings are held, what the outcomes were, but they don't. There's nothing, to my knowledge, that actually tells you whether it's the molecule is a go or no go, essentially.
Got you. Thank you. Another question has been, what preclinical landmines would one look out for with GCPR antibodies?
Clinical landmine with the GPCR antibody. Well, again, normally before an antibody would go into clinical development, the company would have done extensive search for off-target binding, development of anti-drug antibodies, although that's usually not very predictive. So, development of anti-drug antibodies could be a potential problem. Usually with antibodies, you don't get too many landmines, although for some antibodies, it is possible that when you do the tissue profiling or use something like the Retrogenics platform to identify off-target binding to panels of secreted and extracellular proteins, you might hit on something which is a definite no-go for the development.
Thank you, Christine. In the presentation, you talked about assessing differentiation from the competition when looking to in-license molecules. How easy is it to find information about competitor antibodies? I guess many of these will be in clinical development.
Yeah, so you can look at clinical trials databases, so the US.gov. So normally nowadays, most clinical trials are registered and the information is published. If you're a kind of independent researcher, there are databases, there are specialist companies which have access to company databases and drug pipelines and so on. But usually these are not publicly available. You have to pay a fee to access this. And there are specialist search companies out there who, if you don't have an in-house search and evaluation team, then there are companies which you could outsource this work to. So I'm happy to provide a list of possible companies to anyone who's interested.
Great, thank you. Are you seeing any trends as to when deals are being made during the clinical journey? So are you seeing that the deals are being concluded later in the product's life cycle or earlier in the product life cycle?
Well, I think there is, as I mentioned in the presentation, if a product, a drug fails in phase 3 clinical trials, there's the paper which Humira, as I mentioned earlier, which has actually shown that there is a dramatic increase in the number of in-licensings that go on after failure of phase 3. So most companies, they prefer to in-license something which has already received IND approval to move into clinical trials. Obviously, the earlier you go, the possibility of a problem occurring is more, and also the earlier you go, the cost of the in-licensing is usually much lower. But maybe I haven't investigated it enough, but it doesn't, I would say that most of the deals are done in phase 1 or phase 2.
Excellent. Thank you. So the question was, how would an antibody preclinical or clinical process vary from a small molecule, and what are the biggest bottlenecks associated with antibodies?
The biggest bottlenecks associated with antibodies? Well, actually, the processes for developing monoclonal antibody, so I'm not talking about any bispecifics or multispecific antibodies here, they're largely very generic processes. So, they're clearly the process is more expensive than for small molecules. But I think one of the problems with small molecules is that they often they are less specific than antibodies. So there needs to be a lot of development going into them to make sure that they hit their specific target. So if you consider kinase inhibitors, or inhibitors of MMPs, for example, the small molecules target the catalytic domains, and these are actually very, very highly conserved between the different kinases and MMPs. So, the bottleneck for a small molecule, for example, would be getting that specificity and doing all the design of the molecule and then synthesis of the molecule to make sure you can incorporate that into your molecule. And it takes a long time and doesn't always work.
Fantastic. Thanks, Christine. So last couple of questions then. To what extent should a startup expect the company that's performing the due diligence to request additional experiments? And then to what extent do you recommend startups oblige with those requests?
Well, that's a good question, because it happens fairly frequently. It happens fairly frequently that they are asked to do additional experiments. So normally, what might happen is that the big pharma might, allow a certain pilot study to be performed and pay for that. So yes, that can actually happen. Okay. I'm not sure if I've actually answered the question, and whether the startup wants to do it. Well, it depends on whether they actually have the resource available to perform that experiment. Usually it's going to make the deal go forward more easily, but often, it might be a key experiment that can end in a no-go decision. And actually, I've seen that happen.
Okay, thank you, Christine. Final question then. So what would you say the biggest mistake that a startup can make and should avoid making when providing data during due diligence?
What's the biggest mistake? Well, I think it's the biggest mistake that they can make is not actually being prepared well enough in advance to be able to respond to the quite difficult questions that they'll be asked. So it's important to have a good preparation, make sure that you can answer the questions, and if there is some data missing, acknowledge it, and give a reason why you're not providing that data, because obviously the process has to be transparent from both sides. The other thing is to actually try and license a molecule before it's really been fully validated. And I think this happens often with, monoclonal antibodies. They've shown activity in an animal model, but often the company, the startup or the university is not actually aware of all of the humanization optimization in terms of developability of the molecule that needs to take place before it can be ready to go into cell line development and manufacturing. So it's often moving to license something when it's too early.
Perfect. Thank you, Christine. And with that, I'll hand over to Jen.
The muting. There we go. Awesome.
That wasn't an alarm, actually.
Yeah. Oh, okay.
I put my phone on silent.
That's okay. That's all right. Well, thank you very much. That was very informative. In concluding, I'd like to thank Dr. Power for relating her insights into the scientific evaluation and due diligence of antibody therapeutics. And of course, thank Nick Hutchinson for help with the moderating. I also thank you, our attendees, for being here today and joining us for this webinar. An on-demand version will be available in a few days, and I will definitely send a link to it via email to everybody who registered. Please do feel free to watch this and any of our on-demand webinars when it's convenient. With that, I thank you again and have a great rest of your day.
Thanks to everyone. Bye.